In this lecture we are going to discuss the metalloenzyme of zinc, that is a carboxy peptidase. In this lecture we are also going to discuss the structure, function and biochemistry of the carboxy peptidase. Carboxy peptidase is a metalloenzyme of zinc. It can digest the protein as the enzyme is selective. It hydrolyzes those polypeptide in which terminal amino acid has an aromatic or aliphatic branch chain alkane. So let's discuss the structure of carboxy peptidase. So this is the structure of carboxy peptidase. In that the zinc is an active site. The zinc is located in a depression formed on the surface of called ecoenzyme. Close to this depression there is an empty pocket, and zinc is coordinated with two imidazole rings of histidine group of ecoenzyme, and the third position is occupied by the glutamic acid residue, and at fourth position is occupied by a water molecule when the enzyme is at a rest. So the stoichiometry of this zinc in this enzyme is a tetrahedral one. So let's discuss the structure in detail. Structure. Zinc ion lies in deep pocket or lies in a depression formed by called ecoenzyme. Close to this depression there is an empty pocket, or space for the accommodation of substrate molecule. So this is a pocket of ecoenzyme. And in second point, the zinc ion is coordinated to two imidazole rings and glutamic acid residue This is a imidazole ring, and in imidazole ring there are histidine 196 and histidine 69 residues are present in respective imidazole ring, and the third position is occupied by the glutamic acid residue, whereas the fourth position is occupied by the water molecule. So in next step, the fourth position of site is occupied by water molecule. The stoichiometry of zinc. In enzyme is tetrahedral, where the zinc is in +2 oxidation state, that is an active site of this carboxy peptidase. So these are the structure of carboxy peptidase. Let's discuss the reaction of carboxy peptidase. or it can hydrolyze those polypeptides in which terminal amino acid has an aliphatic branch chain alkane or aromatic group. So let's discuss the reaction. In reaction, this is a polypeptide chain and it will hydrolyze this polypeptide chain. And there is a formation of stable product. This is a polypeptide chain. In that, if we use the carboxy peptidase as an enzyme, then what happened? Here, this chain can form the stable product, that is, it can hydrolyze from here and give the stable product, that is, there is a formation of acid and the amino group. So this is a general reaction catalyzed by the carboxy peptidase. This is a hydrolysis of polypeptide chain. So this is the reaction of carboxy peptidase. Let's discuss the classification of carboxy peptidase. The classification of carboxy peptidase is generally depend upon two types. In that, the first type is that the carboxy peptidase can be classified by active site mechanism, and in second type, that is by substrate mechanism. So let's discuss first what is meant by the classification of by active site mechanism. By active site mechanism, in that a metalloenzyme or the carboxy peptidase which has a metal as an active site is known as a metal carboxy peptidase. So the carboxy peptidase which have metal at an active site is known as metallocarboxy peptidase. And the example of this metallocarboxy peptidase, that is a carboxy peptidase, in that zinc is a metal and which is in active site, though, so that's why the metal carboxy peptidase is an example of metallocarboxy peptidase. Now in second type, if the carboxy peptidase which have serine, that is one type of amino acid, serine at an active site is known as serine carboxy peptidase. carboxypeptidase which have cysteine at an active site is known as a cysteine carboxypeptidase. Carboxypeptidase which have cysteine at an active site is known as cysteine carboxypeptidase. So these are the general classification based on the bioactive site mechanism. Now let's discuss by substrate preference mechanism. So let's discuss what is meant by the substrate preference mechanism. Substrate preference mechanism. So in substrate preference mechanism, the carboxypeptidase which have preference to hydrolyze the aromatic or aliphatic chain is known as a carboxypeptidase A. So the carboxypeptidase which have a preference to hydrolyze aromatic or aliphatic chain is known as carboxypeptidase A. Where here A stand for aliphatic or aromatic. In second, the carboxypeptidase which have a preference to hydrolyze the positive amino acid, that is the basic amino acids. So this carboxypeptidase is known as a carboxypeptidase B. So the carboxypeptidase which have preference to hydrolyze positive amino acids positive amino acids, for example arginine, or we can also say that the basic amino acid or basic amino acid is known as carboxypeptidase B. B for basic. B stand for basic. Carboxypeptidase B. So this is a mechanism. This is a classification on the basis of substrate mechanism. Now let's discuss what are the carboxypeptidase. So, apart from hydrolysis of or digestion of protein, there are various functions of carboxypeptidase. So, function. We know that the main and important or vital function of carboxypeptidase is to digest the protein molecule. So, the digestion of protein is digestion of protein is the main function of the carboxypeptidase. Now, we know that the enzyme is selective. It can hydrolyze those polypeptides in which terminal amino acid has an aliphatic or aromatic branch chain alkane. So, the enzyme is selective. The enzyme is selective. It hydrolyzes those polypeptides. It hydrolyzes the aromatic or aliphatic branch chain alkane. Now the third, carboxypeptidase also function in growth factor, wound healing, blood clotting. So it also functioning. It also functioning blood clotting, wound healing, growth factor, then reproduction and reproduction. Now it also helps to mature the protein. So this is also one of the important function of carboxypeptidase, that it also function to mature proteins, to mature proteins. So these are the important functions of carboxypeptidase. Now let's discuss the mechanism of carboxypeptidase. In this mechanism, or this mechanism can act through the displacement of coordinated water molecule of enzyme to the carbonyl group of substrate molecule. So before starting the mechanism, in that mechanism we have to use that type of structure. Instead of, or simply we can say that histidine and one glutamine. Or histidine. So in whole mechanism we use that type of structure instead of this. This is a very rigid, and for sake of convenience we have to use this structure. So let's discuss the mechanism of carboxypeptidase. First, the mechanism of hydrolysis of polypeptide chain So here our enzyme, then this is a chain of polypeptide. In that we have to hydrolyze. We know that the carboxy peptidase can act through the displacement of coordinated water molecule of the enzyme to the carbonyl group of substrate molecule. So there is a displacement of water molecule of enzyme by this carbonyl group, and this is a polypeptide chain. So let's discuss. Here what happened? We have to write the chain here. This is a glutamic acid and this is a histidine. Here, so this is histidine molecule. Now this zinc ion is act through that. Then what happens? There is a hydrolysis of this chain and form again, after the formation of that product, here we can write here also. Our enzyme is regenerated. This is a so this is a general mechanism. Here there is a loss of water molecule. In that the formation of product is like that. This two stable product will be formed, and here at last step there is a enzyme. This enzyme is regenerated. So this is a general structure, function, reaction and mechanism of carboxy peptidase. In next lecture we are going to discuss the next metal enzyme of zinc, that is zinc fingers. Thank you.